Merci beaucoup. I'm very happy today to present our speaker. So Mallar Chakravarty is a computational neuroscientist at the Cerebral Imaging Center at Douglas Mental Health University Institute. He is also an associate professor in the Department of Psychiatry and an associate member of the Department of Biomedical Engineering at McGill University. Mallar received his bachelor degree in electrical engineering from the University of Waterloo and his PhD in biomedical engineering from McGill University. He went on to do a postdoctoral fellowship in Denmark and jointly at the Rothman Research Institute and the Mouse Imaging Center and the Hospital for Sick Children in Toronto. He is interested in the anatomy of the brain. His group focuses on how anatomy changes through development, aging, and in illness, and how the dynamics of the brain anatomy are influenced by genetics and environment. From a more personal point of view, Mallar is not only one of the best researchers I've known, but he is also one of the smartest, funniest, and most generous collaborators I had the chance to work with. So thank you for accepting to be here today, and je te laisse la parole.
Merci beaucoup, puis merci beaucoup pour le vraiment the very generous introduction. Alors, ça fait plaisir vraiment d'être ici aujourd'hui. Je vais juste commencer en anglais, en français, désolé, déjà je me mêle, mais quand même, je vais faire le talk en français parce que je n'ai jamais donné un talk pardon, je vais donner le talk, wow, je me mêle beaucoup. Je vais donner le talk en anglais. Puis tu vois pourquoi, justement, parce que j'ai fait quelques fautes de mots. Puis, mais si vous avez des questions pendant le talk, après le talk, n'hésitez pas à me demander en français, puis je peux tout à fait comprendre ce qui se passe. Alors, so thanks all for coming. It's a real pleasure to be here. There's a lot of friends and collaborators in the audience, so thank you, Simona, for, like I said, for that very generous introduction. It's always been a pleasure for me to be able to be in the richness of the Montreal neuroscience and neuroimaging community. I've worked with many of the professors here on different initiatives. So it's a pleasure to be able to give this talk, and thank you so much for this introduction. And thank you all for being here. It's sunny and beautiful outside, as you can see behind me. And so I hope you'll enjoy basking in the warm glow of your computer screen for the next little while. So I'm going to talk a bit today about the work that we do in my lab, and most of the work in my lab is really dedicated to structural imaging and really trying to understand phenotypic variation in psychiatric illness. And that's going to be, for the most part, the real focus of my talk today. And more recently, as you can see from the title, we do a lot of work across species. We do a lot of work in clinical populations, and we do a lot of work mostly in mouse models, more recently in macaques, which we're not going to be talking about today. And I'll be showing you some of the work that we've been doing in mouse models. And psychiatry presents a lot of interesting challenges in terms of neuroimaging investigation. I gave a talk yesterday at another class, a slightly different one, where one of the students asked me, well, what's one of the contributions of neuroimaging to psychiatry in general? And one of the contributions I can say right now is really its ability to help understand brain circuits at a level that allow for the refinement of different neuropsychiatric disorders, or for the treatment of different neuropsychiatric disorders. So this is work done in Michael Fox's group on treatment-resistant depression, where he's been pioneering this technique called lesion mapping, or causal lesion mapping, where he looks at individuals, or he examines individuals who have the onset of psychiatric illness due to a lesion in the brain. So in this case, he's been looking at major depression, and he uses those lesion locations as a means to refine targeting for different neurotherapeutic approaches. So in his case, brain stimulation is a really key part of his research program. And so he looks at functional connectivity and discerns how can we use that functional connectivity network as a way to define brain stimulations like transcranial magnetic stimulations on the surface of the brain or on the surface of the scalp, or deep brain stimulation targets for electrodes that get implanted surgically. And in doing so, he's been able to show that based on these on the functional connectivity of these lesion maps, he can actually refine targeting to improve symptoms using TMS and deep brain stimulation. So I think this is one of these really beautiful examples of how you can use, he's been working on this for 10, 15 years now, and you can use really detailed analyses of brain circuitry to really refine therapeutic approaches. But for every success story like this, you'll see a lot of complaining stories like this, and you'll see, why is it that we can't find great treatments for most psychiatric disorders? And a lot of these papers will tell you very much the same things. Drug discovery is at an impasse. Not that that's the only way to treat neuropsychiatric disorders. I'm just using this as an example. There's a paucity of novel and valid targets. There's significant etiological heterogeneity in neuropsychiatric disorders. And we don't know enough about the complexity of the brain to actually provide meaningful treatment targets at the individual level. And this last part turns out to be false. You can do, like I said before, these really elegant investigations using lesion maps, using causal approaches where you perturb a system using, say, transcranial magnetic stimulation and look at how the brain responds. So this isn't necessarily what we're doing in my lab, but I do think the approach is similar, and a lot of people have been doing this, where what we're really seeking to do is find indices of brain architecture, look at them in different contexts, and try to get some understanding of how they relate to genomics, cell types of the brain, or even transcriptomics if you use the Allen Brain Institute tools. In my case, in the case of my lab, what we're really interested in is looking mostly at schizophrenia and examining schizophrenia and neurodevelopmental disorders, although there's another branch of my lab that's looking at neurodegeneration. That's work that I've done with Simona over a few years. But I'm going to focus today on these neurodevelopmental disorders. And the real goal is to find a composite way to integrate very disparate types of data to understand something about the behaviors that may emerge in these disorders. And so many individuals have taken what I'm going to call, for the sake of this talk, a bottom-up approach. So we can take, for example, a gene that we know to be related to a specific disorder and try to understand what are the neural phenotypes that differentiate people who carry that gene compared to people who don't. So this is a beautiful review by Claudia Maldonado, who may still be a PhD student with Seb Jacquemont, who's also a investigator at Saint-Justin and Université of Montréal. And what he does here is he tries to look at copy number variations that we know to be causally linked to autism and autism spectrum disorder and depression. I'm just going to get my pointer out here so we can, yeah, here we go. Do everyone see a laser pointer? No. There we go. I can do this. There we are. All right. And what you see here is the mapping of effect sizes for gray matter volume. SA is surface area. CT is cortical thickness across individuals who carry a duplication or deletion of different copy number variants. I'm not going to go into this in too much detail. But the point I want to get across is that red here indicates increase in any of these indices. So here, increase in cortical gray matter volume in the insula, or decrease in gray matter volume in the insula for this particular copy number variant. And blue indicates decrease. So two different genes that are part of maybe similar or different pathways that we know to be causally linked to different neuropsychiatric disorders show completely different effects on the brain when you examine it this way. So one nicer way to do this is to strip away some of the heterogeneity of that we see in pesky humans, because we're all too different and all too difficult to manage, and look at mouse models, right? This is beautiful work done by a good friend and mentor of mine, Jason Lurch, who's currently at Oxford. And this is a really exciting paper looking at the phenotypic diversity related to genes in autism spectrum disorder. So he took 19 mouse models, he examined 19 different mouse models with genotypes associated with autism, and basically plotted the effect size difference between each of those mice harboring one of these mutations and a wild type littermate, and looked at the genetic diversity and then the phenotypic diversity in terms of brain volume across a number of different regions. And it doesn't take long to realize that some of these mice, blue again, is lower effect size, negative effect size. Red is positive effect size. And that, you take two different genes that we say are associated with autism, and you see completely different neuroanatomical variability and neuroanatomical signatures in these subjects. So this is one way in which we can get at the mechanistic insight into these disorders. But the way that we've been trying to do this is actually going from the other way. Can we not start bottom up? Can we go top down? Can we start with brain architecture and look and consider ASD and schizophrenia in a neurodevelopmental context? And can we gain some insight into genomics, cell type, or transcriptomics? And that's going to form the basis of my talk today. And to do so, I'm going to talk about two different species that we've been working with, humans, which we all know and love, and mice. And despite the differences in brain size and brain volume, one of the things that we can do is we can use brain imaging techniques and brain imaging technologies as a way to form an intermediary between these two species. So, and you can use resolution to compensate for the vast differences in brain size and brain volume between these animals. Here I'm going to talk about, in the humans, some work we've been doing in heterogeneity and neurodevelopmental disorders. And on the right, we're going to talk about the impact of, in mice, the impact of exposures on anatomical and behavioral development. And by exposures, I mean risk factors that we know load onto the emergence of neuropsychiatric illness. And I'm going to talk about more about that in the latter half of my talk. So autism spectrum disorder, heterogeneity is considered to be the rule rather than the exception. And this is one of the disorders we've been studying in my lab using large data sets. And even though there's a core set of features and symptomatology that help define autism, there's a whole bunch of peripheral symptoms such as sleep deficits, mood and anxiety disorders, sensory disorders, and so on, that also come part and parcel with many individuals who are in the autism spectrum. What we do know about autism spectrum is that there are a certain number of variables and biological features that you can use to model heterogeneity within the disorder. And these include, for the purposes of this talk, sex, age. We know younger females tend to have a more severe presentation of the disorder. Intellectual disability and IQ are part of the disorder, but you might see someone with a diagnosis on being on the autism spectrum as being 1 or 2 standard deviations above the mean IQ of a population. So you really do see some significant heterogeneity in the disorder. And so this is work that was done by Sashi Bedford, who did a really excellent 2-year master's degree in my lab and published this paper on the heels of it. And she's now a PhD student doing more work kind of really tailored towards autism with Richard Bethlehem and Simon Baron-Cohen in Cambridge. And to do this work, we started off with a very large data set of about 3,300 individuals. And this is something that is a bit dull to talk about, but it's very important to talk about, which is quality control. We spend a lot of time quality controlling this data because in neuroimaging research, we know that motion has a very important signature on the brain phenotypes that we're seeking to understand. And basically, based on a bunch of triage rules, because of the size of the different data sets that we're using, that we're trying to harmonize, we ended up with almost about half our sample, 1,327 individuals, after we excluded individuals for having too much motion or poor quality data. We also had 129 ASD females, which are going to harp on here a bit, mainly because at the time of publication of this paper, This is one of the largest studies with female, like largest sample of females ever studied in neuroimaging and ASD. So we looked at some very simple phenotypes to start, and I'm going to show you how we did the analysis. So here we had multiple sites, about 18 in total, and we used a prospective meta-analysis. So this is stuff that's often done in Enigma. You'll see it in meta-analyses of the literature as well. And basically what you do is you ascertain an effect size, and then you get a conglomerate effect size and a significance level for all of the different phenotypes that we're studying in our group, in this study, sorry. And the important thing here is that we normalized the processing methods, we normalized all the QC methods. So we really had, except for the data acquisitions, we really had one harmonized way of looking at this data. What's very important to see here, based on the heterogeneity of the disorder, is that if you examined any of these studies in and of itself for a very simple phenotype with mean cortical thickness, you would have a very different answer as to how mean cortical thickness is altered in autism spectrum disorder. But by combining all these data, we see very modest effect sizes of ASD versus controls in cortical volume or mean cortical thickness, which we have a fair bit of certainty about. If you look at cortical thickness, so on the cortical sheet, you can measure the thickness of the cortex, so the distance between the gray matter surface and the white matter surface to the peel surface, moving radially out towards the edges of the brain. If you look at the thickness of that cortical ribbon, we see generally increases in cortical thickness in regions that we know to be involved in ASD, such as in the frontal lobes and in the superior temporal gyrus. And this is an effect size map that we see here. What we see is that there are very subtle effects of age. So this increase of cortical thickness is somewhat more pronounced in the earlier stages of the disease. If you do what's called a centered analysis, where we force the intercept of the model to be at a specific age, and that kind of disappears as these individuals get older. So there's this kind of overall normalization of the cortical anatomy in these individuals. And if we look at full-scale IQ as a proxy measure for overall functioning, we see that individuals on the lower IQ of the spectrum show very different increased cortical thickness across most of these phenotypes, and that difference disappears when you get into kind of the mean or the higher end of the function, right? So here we got the brain mapping part right. We didn't get much of a neurodevelopmental context because we just have age really. And we didn't really get into genes and cell tech, which is what I want to do next. So I'm going to show you some work now where we've been looking at another neurodevelopmental disorder, major mental illness. This is work that's ongoing by Alyssa Day, where she's been really interested in trying to map the psychosis spectrum in this neurodevelopmental context. And this is really important because more and more, there's a belief that major mental illness emerges along a continuum from healthy to abnormal. And you can maybe even be able to see signs of it in the general population early on, even if they're not help seeking. And so she was interested in what we're calling the psychosis spectrum here, where one of the end results of the psychosis spectrum, one of the end diagnoses, if you have a frank onset, could be schizophrenia. We know that schizophrenia affects about 3% of society. It's characterized by the emergence of positive symptoms like hallucinations and delusions, negative symptoms such as anhedonia and cognitive impairments. Psychosis spectrum also includes individuals who are subclinical, who are not necessarily help-seeking, but they have distressing psychosis symptoms that we can measure in the general population. And these individuals, if they're showing some level of these symptoms, are at higher risk of eventual transition to a full-blown psychotic disorder. So to do this work, we've been collaborating with Ted Satterthwaite at University of Pennsylvania to work with a really well-characterized cohort where from Philadelphia, which is meant to represent the racial and ethnic diversity of the Philadelphia region. And it started with 1,600 individuals who were non-help-seeking youth recruited from Philadelphia. There's a nice male-female split, and there's a really great progression and representation of the individuals. And I think this is really even doubly important because this data set is actually about 10 years old. And so the PNC group, the Philadelphia Neurodevelopmental Cohort group, was acquiring these data in this kind of representative manner 10, 12 years before we were talking about it in general science. Everyone went through a psychosis spectrum screening, and what they found was that 266 of these youth endorsed psychosis spectrum symptomatology or behaviors. And those are the youth that we're analyzing in the rest of this cohort. So they're not being seen by a clinician. They're just kids who are in the general population who have these type of emerging psychosis spectrum symptoms. And they endorsed greater than one of hallucinatory delusion lasting more than 1 day, an extreme agreement rating profile with the psychosis spectrum rating scale, and an age deviant score of different preclinical psychosis spectrum disorder rating scales. And this is just demographics in terms of sex and racial diversity. So to get a better sense of the neurodevelopmental components that go along with the psychosis spectrum, what we chose to do is try to create an integrated framework for analysis. And in this integrated framework, what we wanted to do was include different morphometric measures so that we can get kind of one composite signature of brain morphometry related to the psychosis spectrum. And just this is really important because we typically only analyze cortical thickness or cortical surface area. However, different morphometric measures like gyrification or cortical folding, they undergo different types of change at different parts of the neurodevelopmental continuum. And so, for example, gyrification, really emerges early on in infancy and changes rapidly in the infant to early childhood period, and changes less so later on in childhood and adolescence. Whereas cortical thickness really imprints during infancy and early childhood, and then this rapid change in sensory motor regions early on, and a much even greater change in association regions later on, kind of during adolescence and maybe even into early adulthood. So we feel that by having multiple indices stacked together, we can get this kind of composite picture related to neurodevelopment. Now, we're not the first people to think about doing this. There's a beautiful paper by Jacob Seidlitz using where they worked on a technique called morphometric similarity mapping. And in their work, what they did is they put together multiple indices related to myelin, gray matter, and surface curvature, and used a connectomic framework where they looked at the connectivity through correlation between different nodes or different regions of interest across the whole brain. And they created a within subject connectome to do that. Now, the challenge with this method is that in the MSN architecture, you can get really big differences with respect to a specific modality, but it's really hard to say in a specific MSN network, what is the major morphometric feature that's driving differences or change within subject. And that's one of the limitations we wanted to address with our work. And so to do that, we use the framework developed by Rehan Patel, who recently finished a spectacular PhD in the group. And he's currently a postdoc at Oxford with Sana Suri and Claire McKay, working on neurodegeneration there. But he developed this technique, which has been a real workhorse for my laboratory since he left. And what we did is we created basically an input matrix comprised of all of the morphometric features that were generated from cortical thickness and cortical surface mapping data generated in this sample. And we stacked them one next to each other in this matrix. And we used a technique called non-negative matrix factorization, which we didn't invent. We just adapted it to our techniques. It was first implemented in neuroimaging by a scientist named Aris Satiris, who's currently in Vanderbilt. But we like this technique because you can decompose this matrix into a series of brain maps, which are represented by this matrix W, and a series of components, which basically map each subject and their contribution onto this series of brain maps. There's some implementation details that I won't go into, but what's really nice is that this method preserves the individual contributions of each metric in each component, unlike the morphometric similarity networks that I showed you before. It gives you really sparse, non-overlapping representation of these maps over the cortex, and it's designed that way. So you don't have to give it specific constraints above and beyond the orthonormal constraints that are within the methodology itself. The other thing you can do is whenever you do a matrix decomposition technique, it's really tough to figure out how many components you should pull out. And Ray worked on this really beautiful set of techniques that allow you to help you understand what is an optimal number of components that can be derived from this data set. And by doing so, the two methods that he developed were the development of cosine similarity by taking multiple granularities of components and splitting the data set into two in random splits of two. And you can look at, do I get stable components out by shuffling the assignment of these two splits of data? And you can correlate that. And then finally, you can also look at the mean reconstruction error. After all, this is a matrix decomposition technique, so the whole goal is to kind of sparsely represent the matrix by just a number of components. And you want to minimize the reconstruction error while maximizing the similarity between the groups. So when we do that, you get a plot that looks like this. The red is the component coefficient, stability coefficient. So 0.6 is quite high, and we get there pretty fast. And what you want to look at are the jumps in the gradient reconstruction error to determine, are we getting a stable number of components out of our decomposition? And by doing so, we identified 8 components for further analysis. It's always a question, there's always debate as to which of these you should be picking. You could have picked 8, you could pick 10, you could have picked 6. But we've looked at these, and I'm going to show the 8 component solution. Roughly all solutions converge nicely on what we show in this 8 component solution. You get a weighting per subject for cortical thickness, surface area, mean curvature, and gentrification index. And this is a normalized weighting across these different indices. And the color here, we just stack the individual's cortical thickness weight here from, in this case, in ascending order from youngest to oldest for each of these metrics. So we have 4 weights per subject across each of the components. And the components look like this. They're nicely dissociable. They spread across the entire brain. And they have, we can basically kind of almost assign them different names, such as prefrontal cortex, visual cortex, and precuneus, for example. So they do kind of seem to localize in very spatially contiguous regions, even though we haven't put that type of constraint on the data at all. We worked with another scientist, Tyler Moore, to look at the behavioral side of things. And so we didn't want to just kind of do a regression against all the different components in the assessments that were done by the PNC group, by the Philadelphia Neuroimaging cohort. So what we did is we looked at a factor analysis and came up, and by looking at the elbow of the curve, came up with a 3-factor solution, which maps very nicely onto what is known about kind of the dominant modes of symptomatology in schizophrenia and frank onset psychosis or schizophrenia. And that would be negative symptoms with a bit of disorganized symptoms in there, disordered thoughts, so thought disorder symptoms, loss of sense of self, and then positive symptoms such as hallucinations and abnormal perceptions. So each individual now is a score in terms of the brain anatomy that we showed across all those different metrics, and now across these symptomology profiles. We're going to take these as our 3 latent dimensions of psychosis symptoms. Sorry, I'm getting a swirling beach ball here. Hopefully this will pass, and it does. Good. We wanted to relate the demographic variables to our brain variables, and we used a partial least squares analysis. So I maybe should have put a slide in that describes how this works for the group. But basically what we're trying to find are linked dimensions of covariation between our demographic and symptomatology variables and the brain variables that we have. On right in B, what we have, the colors represent weights within those specific networks, those specific components that load significantly on this pattern of demographic and symptomatology variables. And so what we see is that in the demographic variables, we see younger age, female sex, European American race, and non-African American race loading very highly with this pattern of anatomy, which I'll describe in a moment, and a lower severity of disordered thought and sense of self in this dimension, right? So it's highly specific to only one of the dimensions that we uncovered in the factor analysis. What's nice though is that it comes with a broad signature of morphometric variables. So for example, we see gyrification index loading in this component 1, this frontal component, and in this kind of opercular superior temporal component. We see differential contributions of cortical thickness. So blue is lower and red is higher in these associated areas in the frontal lobe, mean curvature in the occipital cortex, and surface area in the temporal pole, ventral, and then the ventral parts of the temporal lobe as well. So this shows that there is this kind of broad signature related to multiple morphometric components. And what we wanted to figure out is, are these components developmentally meaningful? So for this, we turned to a collaborator, Avram Holmes, who's at Yale, and we used some maps that he recently developed to look at what's called the gradient architecture of the cortex. So there's been a real push in the functional connectivity literature to characterize brain function as existing on a continuum from unimodal to heteromodal function. And so you can see that here in the adolescence, for example, where blue, or there's less weighting in this gradient in kind of primary sensory areas, such as M1, S1, and in the auditory and visual areas, and higher weighting, for example, in heteromodal areas that do integration of sensory processes. And so to do this, we took these maps as just another representation of the maps that I showed you. I wanted to contextualize these 5 components that we know to be most related to the psychosis spectrum disorder, thought disorder that we saw here. I want to contextualize them in gradients that Avram Holmes and Haoming Dong showed that vary and change across development. And basically, there's more and more separation between the unimodal and heteromodal and the transmodal, the heteromodal regions of the cortex functionally as one goes from childhood to adulthood. And that goes from not being a non-dominant feature in the cortex in childhood to being a very dominant feature of the cortex in adulthood. And so we looked at the distribution of each of these components and how much they're encapsulated by within transmodal areas to the right and unimodal areas to the left. And by and large, these red marks here represent the median of the distribution. And those aren't particularly well differentiated across these components, except for oddly in the visual cortex, which we're not quite sure as to why that is. And we looked at the decile level distribution using a statistical technique called the shift function to look at how different they are in adults versus adolescents or adults versus children. And we can do that across all the different components that we have here. It's a very busy plot, and if I went through all this, we'd be out of time. But what I wanted to get across here is that basically it seems like that what we're trying to uncover is that these components that we've uncovered seem to be rearranging in the context of this shift from unimodal to transmodal across the developmental time course. That's something that we're investigating further. The other thing we wanted to do was characterize these components with respect to the transcriptomic data available brain-wide in the Allen Institute Brain Atlas. And so we're really focusing in on these 5 components that we know to be most related to psychosis spectrum symptoms. And so right now what we did is there's about 1,000 different samples with gene expression data, so RNA sequencing data across the cortex in the Allen Institute Brain Atlas site. And in preliminary analysis, what we've been doing is been looking at the first principal component of variation in that gene expression. What we find is that the major contribution, if we do a partial least squares analysis against this and the weightings of these components, the major contribution, for example, is differentially associated in regions that don't map onto the psychosis spectrum versus regions that do. And we're in the process now of looking at cell type specificity in this transcriptional landscape, as well as enrichment for psychosis-specific genes as well. And so I was hoping to have that ready for today, and I don't. But next time I come, I promise to share. But that's definitely what we're going with this kind of work. So this is our attempt now to start reconciling different levels of brain architecture in a neurodevelopmental context and take this top-down approach to look at least in this case, transcriptomics. And we hope cell types in future analyses. But a lot of this stuff is very much inspired by what people do in basic neuroscience. And a lot of what we do in our animal imaging work is very much inspired by what people have been doing in the neurodevelopmental context in neuroimaging for some time now. And so we wanted to imprint this on the work that we're doing in animal models by looking again at brain development trajectories and trying to then, so to create a developmental context that we can do in animal models and try to figure out how this maps onto transcriptomic data again, or cell types or genomics. And I'm going to talk about some of that work that we've been doing in my lab in the half an hour, 25 minutes 25 minutes that's left. So to do this, we wanted to look at risk factors that may alter development. And I wish we could say we were very timely and we started this set of experiments before the COVID-19 pandemic, but it took us a long time to figure out how to do this. And we started this well before. And the reason I think this is timely, or it turns out to be timely, is that one significant risk factor for major mental illness, such as schizophrenia or autism disorders, autism spectrum disorders, is the exposure to maternal infection during the gestational period. So if your mother, we don't know what's happening with COVID, but we do know that due to flu epidemics or even an endogenous pro-inflammatory cytokine levels due to obesity or stress, that may alter brain development and increase susceptibility for brain disorders such as autism and schizophrenia. And we know that brain disorders in general, such as ASD and schizophrenia, are related to the genetics of the parents, their intrauterine environment, and multiple hits during the developmental period, such as stress, exposure to drugs of abuse, and so on. And that may compound risk for these disorders. So it turns out that in the human literature, you can actually detect differences in brain architecture very early on related to exposure to maternal immune activation or pro-inflammatory cytokines in utero. So this is work done by Damien Fair and Claudia Buss, where they basically took samples of blood and looked at the concentration of interleukin-6, which is a pro-inflammatory cytokine, during the developmental period. And it turns out that the level of interleukin-6 in blood can predict the functional connectivity architecture in babies at 4 weeks old during natural sleep. And that babies born to mothers with elevated levels of interleukin-6 have altered architecture of, in blue here, in black here, the salience network, and in green here, the dorsal attention network. And it's already detectable shortly after birth. So now to see what happens to these kids is going to take, years, if not decades, to figure out, if there's going to be a elevation in schizophrenia diagnosis, autism diagnosis, will they endorse preclinical psychotic symptoms? But animal models present a really excellent methodology for examining this further. And so what we've been doing in our lab is looking at exposure to maternal immune activation during pregnancy in animal models. And you can do this with a viral mimetic known as poly-IC, which is what we used. And poly-IC is a double-stranded RNA, which, when injected in the mother, in the pregnant dam, causes an inflammatory response, which increases pro-inflammatory cytokines throughout pro-inflammatory circulatory cytokines. And these cytokines cross the maternal compartment through the placental compartment and kind of disrupt the careful immune balance that is required for healthy development of the fetus. And so this is work that was really pioneered in my lab by Lisa Guma, who was also a PhD student up until very recently in the lab. What I want to talk about are a series of experiments that she did. Most of all is this paper that was recently published in Biological Psychiatry. Lisa's someone to watch out for. She's now a postdoctoral fellow with Armin Rasnahan at the NIMH and Jason Lurch over in Oxford as well. And so you can imagine that the introduction, the time in which you introduce MIA in the dam, in the mother, in the mouse mother, will have a significant impact on gestation downstream. So for example, if you introduce the viral mimetic at gestational day 9, you might have a very different impact than what will be happening at gestational day 17. And that's mainly because of the types of things that are happening in fetal assembly, type of processes that are ongoing in fetal assembly at that time. So for example, gestational day 9, you may be disrupting things such as neurogenesis and gliogenesis, or at gestational day 17, you may be disrupting neuromigration and apoptosis. So to situate this in our neurodevelopmental context, we looked at those two specific timings of maternal immune activation and examined 4 different time points in the same animal. So we could get a neurodevelopmental trajectory across time that encompasses weaning, so very early childhood, all the way to adulthood in the same animals. And we can get a series of behaviors at 2 different time points in these same animals that we could then relate to their neurodevelopmental trajectories. So it's something that would be very tough to do in a short period of time, obviously, in humans. So we did all this locally from the animal modeling all the way to the behaviors and the imaging acquisition at the Douglas Hospital, at the Cerebral Imaging Center. It's a beautiful place. This grill is what you see behind me now. It casts a beautiful shadow, very poetic. And we have this beautiful 7 Tesla Bruker Biospec magnet that sits right below me that is much smaller than a conventional 3 Tesla magnet, but has beautiful gradients and allows us to do a lot of this work here. And you can analyze this data much like you would human MRI data. In our case, we used a deformation-based mapping. So you can imagine that if you have 2 different mice, mouse brain 1 has a much larger cortex than mouse brain 2. You can find an intermediary phenotype, an intermediate mapping that maps to the average of mouse brain 1 and mouse brain 2. And you can map those deformations back to the individual mice to show that one mouse has increased cortex, increased cortical volume, while the other mouse has decreased cortical volume. And you can do this in a longitudinal fashion where you can do this across mice and then across groups in this sample. And so when you do this, you get statistical maps that look like this. So if you've never looked at the mouse brain anatomy before, I'll just walk you through this. In the back is the cerebellum here. So you're looking at the cerebellum here, and we're looking at the mouse brain kind of in coronal sections going this way, all the way until we get to the olfactory bulbs over here, which is what these little things are down here. This is the mouse hippocampus. These little dark spots are the ventricles. This is the mouse corpus callosum in the frontal lobe. This is the mouse dentate gyrus, for example. And you see, we map this trajectory with a 3rd order spline fit, and these are in the hot colors here are regions that undergo positive change with respect to the spline sign, and in blue are regions that undergo negative change. And so what does this look like in a more meaningful way in terms of developmental trajectories? Well, I'll walk you through a few peak voxels and significant findings. So we're going to focus here on the early exposed, the gestational day 9 exposed animals, because that's where we found the most significant difference between the 2 groups. What we significantly found kind of overwhelmingly throughout, and this is something to keep in mind and look for throughout, and I'm going to harp on this over and over again, is we see this kind of undershoot early on in development, followed by this overshoot, and then a normalization of the brain anatomy later on in adulthood. And this is true in several regions. I'm going to, in a hand wavy, say related to major mental illness, and feel free to take me to task on that later on if you want. In the cingulate cortex, in the striatum, and the hippocampal CA1 dentate gyrus area. So it's a very consistent mapping that we see across all these animals that we've been looking at. We then looked at a bunch of behaviors. I'm not going to walk you through all of them, but I'll walk you through one of them just to give you a flavor of what happened. So one of the things that we looked at was pre-pulse inhibition, which is basically a response to acoustic startle. So an acoustic startle would be, if all of a sudden the fire alarm went off in your building, you would suddenly jump out of your chair because you weren't ready for that. But it turns out if you give a pre-pulse, a priming pulse, a short burst of acoustic activity, the amount of startle that an individual will go through won't be quite as much. This is true, both in animals and in humans. And what's really interesting that in schizophrenia and autism, if you give this pre-pulse, there's a bit of deficit in the dampening and the reduction of the startle. And this is what we see in our early exposed animals. There's reduction in the amount of startle. They don't go through the expected amount of reduction. They still startle quite a bit relative to the late exposed group and the early and the saline group. This is true across decibel levels, but again, pretty much disappears in adulthood. So now what I showed is we have a tremendous amount of information at the single mouse level, where we look at how they develop over time, how their behavior develops over time. And what we wanted to do was bring those together to create a meaningful phenotype of disordered brain development and behavioral development that speaks to variation in the individual mouse. And the way we do this is by using partial least squares. And so here we use just one time point in adolescence with their behavioral data and their brain data. You can correlate these two to create a cross-correlation matrix that is then decomposed as single valued composition, which gives you a brain map and a behavioral map, which is connected by a singular value. So what this looks like, in principle, is a latent variable that connects the disordered brain development to a behavioral phenotype. So, and it shows that these mice basically exist on a spectrum. They're not disordered or disordered, but like humans, they exist on the spectrum, both in the brain anatomy and in their behavioral phenotypes. And this is one of the first demonstrations that you can actually use this approach in animal models. And so in red, you see the regions that undergo this disorder, and they're associated with decreased locomotion, improved social behavior, and improved sensory motor gating on the pre-pulse inhibition test. But these blue areas show the opposite patterning in these animals. And if you look at the individual brain and behavior scores in pink again, you see there's a significant difference in the mice who undergo early exposure to maternal immune activation than the other mice. So what we wanted to do was use the neuroimaging as a roadmap for looking at transcriptional signatures related to this disordered brain and behavior development. And we did this with Rosemary Baggett, a very talented postdoc in our lab, Verdana Svetkowska and Pedro Bourguignon, where we looked at the anterior cingulate, the dorsal hippocampus, and the ventral hippocampus. I think for time, I just won't go into this. What we see is we see differential gene expression at the time of sacrifice of these animals, which isn't heavily related to the anterior cingulate or the ventral hippocampus, but we see a significant differential gene expression signature in the dorsal hippocampus, where we see here in blue, the downregulated genes, here in red, the number of upregulated genes. And if we looked at, and we see a significant contribution of NF-kappa beta, which is a gene which is well known to be involved in neuroinflammation. In the ACC, we see genes that are related to IL-1 signaling, which is involved in both innate and adaptive immunity. In the ventral hippocampus, we see genes that are related to IL-17 signaling, which is another pro-inflammatory cytokine, which has been previously shown to be critical, a critical cytokine for inducing MIA deficits, and apoptosis as well. I'm going to jump to what we find when we do kind of a more broad enrichment type of analysis. And so in red are upregulated kind of behaviors and genes that we see from our enrichment analysis, and in blue, downregulated genes and behaviors. And we see this incredible contribution of the signaling pathway called FGF, which is classically involved in tissue repair and is critically a signal protein that's involved in embryonic brain development, the dorsoventral patterning of the spinal cord, and supports normal cortical gyrification. We also see autistic behaviors significantly enriched, and microRNA expression, which have been known for a long time to be, or more recently to be involved in major mental illness as well. So I'm just going to go to this FGF pattern that we see and why it's important. So like I said before, it's really involved in forebrain patterning early on in embryogenesis. It's involved in cortical gyrification. So in this work done by manipulating actually the ferret brain, you can see its contribution to cortical gyrification. It's involved in the dentate granular layer cell maturation and excitatory synaptogenesis, excuse me, and cortical asymmetries, which we know to be altered in autism and schizophrenia. So I'm going to just quickly go through what we decided to do after this, which is to further contextualize this neurodevelopmentally by looking at early stages of brain development. So because we're using mouse models, what we can do is we can actually look at fetal brain development using the same two time points that we used before, gestational day 9 and 17. And we did this with the help of Jason Lurst when he was over in Toronto by using really sophisticated technology, MR-based technology, where you can scan these animals, the embryos overnight, and look and get brains at and whole body scans at 40 micron resolution. And when you average these together, you get this beautiful mapping that looks like this. If I could get you there, that's the whole brain of the whole body of the embryo. And here you see the brain there. And we see some familiar brain regions, such as the prelimbic area, which showed up early on in GD9 exposure, the septal nucleus as well, and the subventricular zone, including the caudate and putamen area, as well as the hippocampal and dentate gyrus. The GD17 alters brain volume to a much greater extent. We see kind of widespread increases in brain volume in the septal nucleus, the pallidum, across the cortical plate, and the corpus callosum, and the hippocampus, and the central median nucleus. We worked with Marie-Ève Tremblay and Maud Bordelot, who were previously at Cerveau and Laval and now are now at University of Victoria. And we saw an increased proliferation to a certain extent in dark glial cells using electron microscopy, which allows us to probe these embryos at the nanometer resolution, and dark neuronal cells, particularly in the late group. And we see increased apoptosis in this early group, which brings us closer to our stated goal. When we look at neonates, using a very similar design, looking at PND8 when they're still with their mother, weaning, and they haven't weaned yet, we see very little actually in terms of neuroanatomical differences. And this is something that we're still actively looking at. But it's very interesting that we actually see very little in this context. So altogether, what this allows us to do is really situate this one risk factor for brain development in a full neurodevelopmental context. We can look at cell types related to embryogenesis, which is what we did, and relate them to the brain phenotypes that we get from MRI. We can look at these early life behavioral, which I didn't talk about today, and MRI-based signatures, and this full neurodevelopmental context from childhood to adolescence, which allows us to look at brain development trajectories, relate them to the transcriptional architecture of the brain in both GD19 and GD17. So in conclusion, what I'd like to say is that I think for us, we think that there's this increased benefit of using translational assays like neuroimaging to gain insight into psychopathologies. I think it provides us a way to get better links with genetics, environment, phenotypical expression at the individual level, and look at their ultimate impact on psychopathology. And we think that this may be a key way forward using basic and clinical neuroscience to parse heterogeneity across neuropsychiatric disorders, and maybe even ultimately promoting pathways towards clinical medicine. But I think it gets us one step closer to getting to the stated goal of kind of this aggregate methodology for looking at how all these different architectures may work together. Finally, I just want to thank all my wonderful collaborators from the MNI, from McGill, from University of Victoria, and IMH, Oxford, and Yale. Of course, my family, who let me work and travel and but still, let me hang around and try to have some fun with me. My son Panaki and my wife Rachel McCabe, my funders, and finally my wonderful lab, without whom a lot of this work wouldn't be possible at all. I'd specifically like to thank Gabriel Devenny, who's been and Daniel Galino, who've been working with me for 7 years as research associates, and just do a wonderful job at helping with the students and myself try to understand some really sophisticated technical aspects of this work, and without whom a lot of the students and myself wouldn't be able to show off this work that you see today. So with that, I know I'm a little late, I apologize, but I'd be happy to take your questions. Thank you very much.
Thank you so much, Mallar. Merci infiniment. Même, tu dis que tu as pris un peu running a little late, but non, c'était tellement intéressant. Sérieusement, je n'ai pas vu le temps passer. Fait que merci beaucoup, beaucoup. Je crois qu'il y a plein de gens qui applaudissent aussi. Donc, je voudrais voir, je vais ouvrir juste le panneau des participants pour voir. On va prendre une période de questions si ça te va toujours, Mallar.
Absolument, oui.
Parfait. Donc, je vais juste ouvrir mon... Et Cassandra, Catherine, si vous voyez des questions ou des mains levées que je ne vois pas, n'hésitez pas à m'aider aussi. Donc, je vois une main levée. Attendez, là, ça se met à jour, fait que là, ça disparaît. Oui, donc il y a Myriam qui aurait une question.
Yes, so thank you very much for the talk. It was very interesting. I have more of a technical question. I'm not sure I understood well for the non-negative matrix factorization. How were the components selected and extracted?
So we have, which part of that was challenging? So the input part is that you have all of these kind of cortical features that go as input. And then the components are extracted by looking at two features. One, it's basically a split half stability analysis. So you do multiple splits of the data, and you look at how well can you recapitulate basically the components that we see over and over again. How close are they to one another? And you can do that across multiple granularities. And so you want to get to a point where there's a high similarity, and then you plateau, and there's not much benefit to getting more components. And then you can look at the reconstruction error, because this is basically data decomposition techniques. So you can do kind of the equivalent of Procrustes rotation to see what is the difference between the reconstructed data and the original data set. And you want to minimize the amount of error there. So you kind of use these two metrics to do component selection. Does that make sense?
Yep. Thank you very much.
Donc, n'hésitez pas à lever la main si vous avez une question. Ah, il y a Simona qui a une question. Karim après aussi.
Merci beaucoup, Mallar. Écoute, j'ai une question par rapport donc l'hétérogénéité et les caractéristiques que tu vois au niveau structurel très tôt dans la vie. Tu penses qu'ils ont une relation possible aussi par rapport au profil cognitif qui est très différent entre une personne et l'autre, aux différentes maladies aussi neurodégénératives reliées au cerveau qu'on peut développer plus tard dans la vie? J'imagine qu'on n'a pas assez de databases pour avoir cette perspective-là, mais quel est ton insight sur ça?
I mean, les données qu'on a vues ici, c'est que effectivement, il y a cette architecture, c'est vraiment lié dans la hétérogénéité qu'on voit au moins pendant l'adolescence. Vraiment, parce que Puis aussi, qu'est-ce qu'on a vu? Je ne sais pas si c'était apparent, mais c'était aussi différent par rapport à la race. Mais pour ça, je ne veux pas dire que les races sont différentes par rapport à leur architecture, mais leurs expériences par rapport au stress, au niveau de leur environnement, c'est clairement différent dans les États-Unis, dans une grosse ville, par exemple. Alors, moi, je pense qu'en totalité, toutes ces expériences en influence sur la croissance, la maturation du cerveau. Puis cette hétérogénéité, c'est facilement peut être lié aux signatures cognitives cliniques. Puis je pense que de plus en plus, on peut juste caractériser ça si on fait vraiment, if we really improve, si on est capable d'agrandir le spectrum des gens qu'on étudie dans nos études, de mon côté. Je ne sais pas si ça c'est...
Ouais, ça répond. Thank you.
Thank you.
Karim, tu peux y aller? Si tu as encore une question.
Oui, merci. Well, thanks, Mallar, for an amazing talk. The amount of work is just fascinating. It's mind-blowing and yeah, very, very impressive. I have a question, well, two quick questions. So one is out of curiosity, the link between structure and function in terms of connectivity studies. There has been sort of diversity of results. Some papers have found a strong link. Others have found, like, anti-correlation between the two things, or lack of association. And so I was just curious, your take on this. If we look at, most of this has been done with like functional resting state data and looking at how those results connect what we see from structure on MRI. And I was wondering whether, for example, you think that electrophysiology would have a better chance of better tapping into that function structure relationship, or is that a wrong way to approach the question?
I think that's somewhere that we're well situated to look at. So we like to be able to reconcile some of these debates kind of with this combination of kind of this species, this multi-species approach that we're taking. So I agree with you. I don't know. I mean, even for the structure of function homologies, I mean, so much depends on even how you characterize function and how you characterize structure. And even within either of those, whatever connectivity means to you, it could be a gradient, it could be a graph theory network, it could be an ICA-based DMN, for example. One of the things that we're looking to do is we have a recent experiment where we've been actually looking at kind of treatment response to different to new drugs that are being used in neuropsychiatric illness. It's not, we don't have the data here, but in those mice, we have two phenotypes. One is the structural, and one is actually a functional imaging paradigm, which we've worked really hard to optimize for animal models. Now, the beauty of animal models is we can actually look at, one, our changes in anatomy due to some sort of, at least in part, some sort of activity-dependent plasticity or experience-dependent plasticity. And to get at that, we can actually do simultaneous imaging of specific nodes in the brain using calcium while doing MRI. So we can do that with the function and with the structure. And so that is a technology that we're working on now. So I don't have an answer for you yet, at least with our data. I'd only be able to tell you what other people do, but I'm hopeful that we can. I'm quite agnostic to this. I don't have a camp that I'm rooting for. I'm not team homology versus team lack of homology, but I am, I am in support of studying it further in this way. And I think this gives us some ways to do it.
Thank you. I mean, the temporal, the variation in time is an important component, because you touch upon this now in your answer. So, for example, if we assume that structure changes, I mean, it's a wild assumption, but maybe is less variable over time than, for example, functional resting state. Maybe it isn't. But if these things are very closely related, you'd maybe expect both of them to vary in the same amount. But if across weeks, so resting state fMRI studies or even resting state MEG shows some degree of stability, but there's also quite a lot of variability over time in the connectivity matrices, for instance, in resting state data. Do you think that's on the same scale of changes that we see in structural connectivity?
The functional stuff that we have that we've worked with locally, it's rock solid, scan test retest. It's your 95% kind of test retest reliability for sure. What we do find is we can detect significant changes in relatively reasonably sized cohorts of 15 to 20 animals, due to a stressor or due to a environmental exposure in 5 to 6 days, is, temporally is nothing. I was surprised when we captured that, but really, 5-10% changes in brain volume locally in 5 to 10 days, which isn't, they're not driven by outliers. It's a pretty, consistent phenotype. So how that relates to functional reorganization, which you would imagine would be happening still on a timescale which is much quicker, we'd hope. That's the open question to us. And I think that's where we're hoping to go. But at least one of these modalities is changing over time in a relatively short period of time. So there should be something we can capture across both architectures. That's somewhere we hope to figure out. That's a great question.
And in humans, presumably you see things changing at the same scale? Like, would you expect volume changes in the space of a few days? Is that what
reported volume changes within day, based on training or stimulus. Over a few days for sure, based on intensive training, volume changings for sure. That I think is real. But within day, I have my doubts, but it's still something, that's an open question for sure.
Before and after lunch.
I can tell you how I feel before and after lunch.
Depending on what you eat.
Yeah, exactly.
Thank you.
Thank you.
Merci, Karim. Je vais regarder. Peut-être, si vous avez des questions, n'hésitez pas à lever la main. J'ai peut-être une petite question aussi par rapport à la cohorte, le Philadelphia Neurodevelopmental Cohort.
Oui.
Quand je regardais, c'était environ, je crois que ça donnait près de 20% des enfants. C'est comme des gens qui avaient pris dans la région de Philadelphie pour essayer d'avoir des ados ou jeunes adultes représentatifs de la population générale. C'est ça qu'ils faisaient?
Oui, c'est ça.
Okay,
alors, ça commençait avec 1600, mais nous, on a seulement utilisé les gens qui ont des symptômes qui apparaissaient par rapport aux critères que j'ai donnés. Alors, il n'y a pas tous les gens qui ont des troubles psychotiques ou des symptômes psychotiques qui sont détectables. Il y avait juste une 20% de la cohorte qui avait ça.
OK, puis c'est parce que j'étais surpris justement que ce soit presque 20%. Est-ce que c'est... je ne suis pas en psychiatrie, moi, donc je ne connais pas toutes les prévalences et tout ça. Puis là, on parle du spectre de la psychose. Est-ce que c'est quand même les chiffres, pas de prévalence parce qu'on parle de spectre, là, mais est-ce que c'est autant que ça, presque une personne sur 5 va être sur le psychosis spectrum, ou c'est dans cette cohorte-là, c'est quand même assez élevé? Non,
effectivement, une personne sur 5 va avoir quelque part dans sa vie quelque chose qui ressemble à un symptôme de psychose. Alors, un exemple, c'est si tu n'as jamais été chez toi, puis tu étais toute seule, c'est une longue journée, puis tu es fatigué, tu entends quelqu'un qui a dit ton nom quelque part. Ça m'est arrivé, c'est certain. Absolument pas trop souvent, mais ça arrive. Mais il y a une 20% de la population pour qui c'est pas quelque chose qui est it's not diagnosable as a disorder, but it's certainly in this class of subthreshold symptoms.
Oui, c'est dans le spectre. Et non, c'est pas un trouble clinique, mais mais c'est élevé quand même pour des adolescents. J'étais étonnée. Ouais. Mais ce n'est pas mon expertise. Alors, OK. Puis, j'ai peut-être une autre. Je vais juste vérifier s'il n'y a pas, parce que je ne veux pas voler le temps de questions à d'autres personnes. Je vois qu'il n'y a pas d'autres. Mais j'ai une autre question qui est vraiment comme plus globale, mais je suis super impressionnée là, par le type de travail que tu fais. Je trouve ça, c'est super intégratif. Puis tu le présentes vraiment très bien. Là, c'est facile à suivre. Et puis, j'aime ça, ces études-là, un peu comme tu as parlé aussi de Sébastien Jacquemont, des études aussi très intégratives avec beaucoup de types de données. C'est fascinant. Mais tu disais souvent, c'est des broad signatures qu'on va trouver. Je crois que tu citais un papier au début. Puis, je me demandais, vu que justement, c'est c'est pas spécifique nécessairement ce qui est trouvé comme signature. Je vois tout à fait l'importance de faire ces études-là, puisque ça apporte sur le plan scientifique, là, vraiment, c'est immense. Mais je me posais plus, la clinicienne en moi se demandait comment qu'on va pouvoir, selon toi, là, dans un avenir futur ou pas trop lointain, comment on va pouvoir utiliser ces études-là pour guider nos traitements ou pour faire la prise en charge clinique? Parce que ça reste tellement encore pas spécifique que comment on va pouvoir utiliser ça concrètement?
Moi, j'utiliserai l'exemple que j'ai donné au début avec le functional connectivity dans la dépression. Parce que cette ligne de recherche, ça a pris McFox presque 15 ans pour arriver là, où il est confident qu'il y a une confiance qu'il peut utiliser ces réseaux-là pour faire pour cibler des traitements de TMS. Alors là, dans une façon, c'est vraiment direct. C'est vraiment un traitement causatif. On donne une charge, on donne une stimulation, puis ça change le réseau, puis ça change le symptôme, puis c'est observable. Mais là, on sait qu'il y a un spectrum de génétique, des transcriptions, des réseaux cérébraux qui se présentent dans une population, puis même dans les souris, que je trouve vraiment impressionnant qu'on peut voir ça. Alors, je pense que c'est plus de recherche. On commence à juste... Je pense que pour nous, vraiment, c'est l'idée de faire ce lien intégratif. On est vraiment dans le domaine de juste... Il faut qu'on le fasse. C'est intéressant d'un point de vue scientifique. On pense quelque part, ça peut aider precision medicine, des affaires comme ça. Mais vraiment, si on donne les outils de Allen Institute comme un exemple, ce papier-là, c'était publié en 2012, où il a donné toute la transcription cérébrale dans les cas. Il a pris une autre 5 années pour le premier papier qui a utilisé ça en intégration avec les données d'imagerie. Puis maintenant, il y a des débats qui se lèvent de comment utiliser cet outil-là en conjonction avec l'imagerie. Alors, je pense qu'on est vraiment dans le methods development phase. On n'est pas dans on a les réponses par rapport à toutes ces données-là. Mais avec les méthodes que, par exemple, Karim, il travaille dessus, nous-mêmes, les autres à Montréal, les autres ailleurs, je pense qu'à un moment donné, on va faire des convergences par rapport à des signatures, par rapport à des techniques, par rapport à des méthodologies. Mais ça va être après ça qu'on peut utiliser ça dans une façon clinique, je dirais. Maintenant, je pense qu'on est vraiment dans pas de data mining comme tel, mais vraiment dans est-ce que les outils qu'on utilise, est-ce qu'ils font de bon sens par rapport à qu'est-ce qu'on aimerait découvrir. So that's like a real non-answer to your question. Give me more time and maybe we'll give you some answers.
Peut-être pour faire du pouce sur ce que tu disais, est-ce que tu penses que dans notre carrière, je n'ose pas dire de temps, là, dans 50 ans, mettons nous, là, est-ce que tu penses qu'on va avoir la chance de voir, par exemple, pour des enfants chez qui on suspecte un trouble du spectre de l'autisme, chez qui on suspecte un TDAH, chez qui on suspecte un trouble neurodéveloppemental, penses-tu qu'on va avoir assez de données, des bases de données assez grosses, avec des âges assez larges, puis assez d'hétérogénéité dans nos bases de données, pour être capable de dire sur un scan, l'enfant est à risque de tant de pourcents de développer un ADHD ou penses-tu qu'on va arriver là?
Et ça, c'est une super question parce qu'en fait, j'avais une discussion par rapport à ça aujourd'hui. Par exemple, je suis parti d'un réseau qui essaie de faire des avancements dans Youth Mental Health, comme en gros. Puis un des problèmes, c'est la représentativité des échantillons. Comme si on parle des travaux qui ont été faits au Douglas, la majorité de c'est vraiment des patients, des clients qui viennent d'une catchment area. C'est vraiment les gens qui viennent de près d'ici. Alors, c'est un type de client. Si on va vers les États-Unis, parce qu'il n'y a pas il n'y a pas il n'y a pas de système de santé qui est centralisé. Les clients, ils sont vraiment, d'habitude, si on parle de psychose, c'est d'habitude des gens qui ont été hospitalisés. Ce n'est pas les gens qui agissent dans la communauté. Alors déjà, on a un manque de représentativité parce que ce qu'on parle early psychosis ici, ce n'est pas early psychosis pour les autres groupes. Alors, c'est vraiment d'aligner d'un spectrum de qu'est-ce qu'on parle pour voir. Ce n'est pas juste la quantité de données, parce que c'est facile d'acquérir beaucoup de données qui ne vaut rien, en fait. Mais c'est plus difficile de faire que mes données à moi peuvent communiquer avec les données qui sont venues des États-Unis, d'Europe. Même en Europe, nous, on parle de spectrum ici, beaucoup de dimensions, par exemple. Je parlais avec une collègue en Suisse, à Zurich, puis lui, il ne fait jamais ça, puis je lui demandais pourquoi. C'est parce que les cliniciens, ils n'aiment pas ça. Ils n'aiment pas ça, et ils veulent les diagnostics clairs, précis, même s'ils n'existent pas. Alors, pour lui, c'est difficile de parler et travailler avec eux autres parce qu'ils ne parlent pas ce langage-là. Alors, vraiment, même ça, il y a des débats par rapport à ça. Alors, j'espère que oui, dans notre carrière. Mais moi, je n'ai pas pensé que pour la dépression, pour Major Depression, on va avoir des signatures si précis qu'on a maintenant. Parce que le problème que tout le monde parlait, c'est qu'il y a tellement d'hétérogénéité dans la dépression qu'on ne peut jamais avoir un ou deux traitements qui marchent bien. Mais je pense qu'il y a clairement des traitements qui marchent maintenant. Il y a aussi des traitements comme intéressants, comme kétamine, comme psilocybine, qu'on commence à rechercher. Alors, vraiment, cette spectrum-là, ça m'intéresse beaucoup aussi parce que c'est vraiment quelque chose de nouveau. Il y a des évidences vraiment claires que ça marche. On ne sait pas pourquoi, mais ça marche. Alors, et ça aussi peut nous donner des indices de qu'est-ce qu'il faut qu'on fasse dans l'avenir aussi pour comment ça marche. So, j'espère que oui. L'optimiste dans moi a toujours l'espérance, mais remains to be determined, I think. But that's our job.
Vraiment, ouais. Parfait. Oui, vas-y. Est-ce que tu allais dire autre chose?
Moi, non.
OK, je pensais que je t'avais coupé. D'accord. C'est les joies du Zoom. Oui, Karim?
Oui, j'ai une dernière question très rapide parce que c'est un truc qui m'intrigue un peu. C'est en schizophrénie, on parle souvent des déficits en cognition sociale.
Oui.
Et c'est quelque chose, moi je me demandais dans le nombre d'études que vous avez fait dans le laboratoire, en regardant aussi la génomique, mais aussi les changements de volume et tout ça, est-ce que dans ce que tu vois, il y a des insights qu'on peut gagner de ça par rapport à vraiment cette composante cognition sociale et comment les altérer? Éventuellement, est-ce que ça peut nous renseigner aussi sur la cognition sociale chez les sujets contrôle normaux aussi? De regarder comment c'est...
Oui, on a vu ça dans les souris. On n'a pas regardé ça dans... C'est pas quelque chose qu'on a vu explicitement dans l'échantillon humain, mais effectivement, c'est quelque chose qui est vraiment important parce que ça, c'est une des déterminantes de recovery pour beaucoup de monde. Alors, c'est vraiment quelque chose qu'il faut qu'on regarde plus. Absolument, effectivement.
Okay.
Et c'est pas quelque chose à regarder.
C'est ça. Tu penses qu'il y a un lien avec vraiment qu'on peut identifier avec les gènes? Donc, ce que tu vois chez la souris en lien avec la cognition sociale, il y a une composante.
On n'a pas regardé. Il faut qu'on fait des manipulations plus profondes avec les gènes parce que c'est une direction qu'on essaie de voir avec DREADS, avec pas encore, même orthogénétique, mais on n'a pas fait ça encore. Non.
OK. Thanks.
Merci beaucoup. Donc, je vois là, le temps qui file, et j'imagine que tu as d'autres choses à faire un vendredi après-midi. J'aurais beaucoup aimé qu'on puisse être en personne pour boire un verre de vin tout le monde ensemble, ou une bière, puis jaser autour de ça, mais on va se reprendre. Et donc, merci vraiment beaucoup pour ton talk. C'était super bon comme d'habitude. Donc, merci. Et puis merci aux gens aussi qui étaient là pour écouter. Donc, je vous souhaite une très, très belle fin de semaine.
Toi aussi, puis merci beaucoup pour l'invitation.
Merci.
Au revoir.
Bye bye tout le monde.